The first is Dr. Malcom Low, who is looking at the role of Magel2, the Magel2 gene again, in the developing hypothalamus. And Dr. Low is really, he's kind of a giant in the field of understanding appetite and metabolism and neurobiology of appetite. He has been looking at brain development in the mouse as to understand this process better, and has found as part of that other work that Magel2 is expressed in these specialized neurons that are important for controlling hunger. And so he is using some new technologies that they have available in his core facilities to study how the genes are coordinately expressed in neurons. And that, he believes, will provide some new insight into the function and these appetite circuits. So the study goal here is to use some cutting-edge technology, this single-cell RNA-seq analysis, which looks at one cell at a time in the developing brain to understand how Magel2 expressing neural circuits develop, and what happens when Magel2 is missing, how that circuitry gets altered. And we're excited about this because Dr. Low is a highly regarded international expert. He's done groundbreaking work on understanding how the brain controls hunger and appetite. His group has a lot of experience in some of these technologies, so it'll be relatively easy for them to apply to PWS, to the PWS models. And the study is going to fill an important knowledge gap in how Magel2 is expressed during development, which is probably quite important for, how the hypothalamus ends up in PWS. So the long-term goal here is to identify neurocircuits that control food intake in PWS and to identify targets for drug development.